Well, hello, human biology class. Coming to you 7 o'clock Tuesday night after the lab exam. I've got them all graded. They're in your POs. My assumption is that when you actually view this will be after we've completed talking about red blood cells, which will be on Wednesday. Just got back from Devani, so if I got some hoagie schmutz hanging from my face somewhere, just forgive that, please. Anyway, white blood cells, the last piece of the circulatory system. I don't know how long this will take, but we'll give her a go. So, yeah, white blood cells, like you see in the image right here, are white. They have that distinct appearance when they are not stained like we stain them in lab, and stand in stark contrast to the red blood cells that you see here. There are some very obvious distinct differences here. You'll notice, first of all, the appearance of the surface of them is very rough. There are lots of bumps on them. Not all white blood cells have those, but a number do. We'll talk about them just a second here. They have a nucleus, and so they are capable of producing proteins, for instance, among other things. They have no hemoglobin, so they can't carry oxygen. So we talked about with red blood cells how it's helpful if the nucleus is out of there, there are no ribosomes, mitochondria, so it's not going to spend energy or use oxygen to create ATP like might happen if there were a mitochondria there. And so there's more space to carry hemoglobin, hence more space to carry oxygen. But it also means they can't heal themselves. Red blood cells can't heal themselves if damage occurs. Like I showed you in the capillaries and the frog webbing, they lead a tough life. They have to turn some pretty sharp corners and things of that sort. So white blood cells are different. They can repair themselves. They can live much longer. In actuality, there's far more white blood cells outside of the circulatory system than is actually inside the circulatory system. And I'll make that case here just a second. Their function is chiefly to fight infection. And so if we were in class right now, I might ask you what would be something that we might be infected with. In fact, I probably will ask you that again when you get back from spring break and we start talking about the lymphatic system. And I'm guessing some of you would come up with some fairly straightforward ones like bacteria and viruses, and you'd be right. Somebody might also say parasites. If you've ever been camping, don't drink the water because you might have giardia in it and give you gastrointestinal distress. So there's lots of things to fight, even our own cells that have gone awry, cancerous cells. White blood cells sometimes will target those for destruction. And so they have a big job to do. Again, another thing you'll hear me repeat, it has been estimated that in our lifetimes, we will encounter over 1 million different pathogens. 1 million different things that our body has to defend us against successfully each time. Because if our body fails, we die. And so this is a pretty important system, pretty amazing system. As your notes say, white blood cells may live from just a few days to many years. They have that possibility. They can kind of hunker down out of sight in your body, even outside of the circulatory system, as I say. They are not then subjected to the slamming and banging around in the vasculature, and they can live a long time. Kind of think of them like a sentry or manning a guard post to defend you against pathogens that might try to enter somewhere. They don't always have to be moving around to do that. They can be standing guard at particular points. And if you think about where they might be standing guard, where are places that pathogens, bacteria, and viruses can gain entry into our body? Certainly the respiratory system, through the mouth, the digestive tract, airway, as I mentioned. Anytime you get a cut, lots of places that we might want extra guards on duty because they're good places for pathogens to get in. That's where you'll find white blood cells as well. Also, we're going to be found in the lymph system and can enter tissue, as I mentioned. In the lymphatic system, that's the next one we're going to talk about. They play a very important role. In fact, some white blood cells really mature in parts of the lymphatic system. And so there's a real common ground between the circulatory system and the lymphatic system. So they can enter tissues, it says right here. And I want you to think about, well, your textbook has a good image. Oh, did I just lose it? No, there it is, of how that happens. So this is a blood vessel, and here is a white blood cell, and there are openings. We already talked about openings between cells. You know the array of different types of openings that exist, from gap junctions and aquaporins being the small ones on up to bigger ones that white blood cells can squeeze through. And so this is a good artist rendition of it. But I want you to do right now is just pause this video, go to Moodle. In the link, there is a link in Moodle. There's a link to octopus squeezing out of a bottle. Just check that out. It's like 43 seconds. And really, that's a good visual representation of how white blood cells get out of the circulatory system and actually enter tissues. Good image to have in your mind when I talk about that. Okay, so they enter tissues. They can be either granular or agranular. So if we look at this is an artist rendition I showed you before in lab of the different types of white blood cells. There are 1, 2, 3, 4, 5 of them. I'm lumping the lymphocytes together because we're gonna subdivide them in the lymphatic system. We're gonna call some B lymphocytes and some T lymphocytes, depending on where they mature. But they're both lymphocytes. So monocytes and lymphocytes, they don't have a rough exterior, really, a grainy exterior. It's these three right here that do. The phils, okay? Eosinophil, basophil, and neutrophil. Neutrophil is the one that you saw most commonly in your blood slide. So if we go down and look at what they actually look like in a blood slide, here we have a collection of them. This is an eosinophil with that red-orange color. This is a basophil, that blue-purple color. And this is a neutrophil. You can hardly see the graininess on a neutrophil, but it's there. So 3 granular white blood cells, neutrophils, eosinophils, and basophils. And then 2 agranular types, monocytes and lymphocytes. They don't have that grainy exterior to them. I'll just slide this over. Okay. Oh, I guess maybe I'll slide that over. So if we look at those, the granular and agranular thing, I think is pretty straightforward. I think I've made the point about that. The artist rendition of these I kind of like. It shows the graininess of the neutrophil. Also that funky nucleus on all 3 has a big parts, skinny parts. That's typical of the nucleus on a granular lymphocyte, a granular white blood cell. So let's look at the granular ones, the phils, if you will. Neutrophils, this is the most common kind of white blood cell. And they're basically bacteria eaters. Now, I'm going to have you pause the video again, or this video again, go back to Moodle, and so that you have an impression, I want you to watch this one. This is also under a minute. And now in this case, it's an amoeba eating a couple of paramecia, but really, this is a good image to have in your mind of how a neutrophil works. They'll encapsulate a pathogen. They'll dump digestive enzymes into the vacuole that they create around that pathogen and digest it, just like you see the 2 paramecia getting digested by the amoeba. Eosinophils, eosin, that's a stain, it's a common stain, this sort of red-orange stain, that's why the name, eosinophils. They combat allergies and parasites. There's other things they do, but those are kind of the main things that they do. So if for instance, you have an allergy to something and you made a blood slide, you would likely have a much higher percentage of eosinophils while you were fighting off that allergy than if you weren't fighting off the allergy. That's how your body would respond to you having an allergic reaction. But you can see they're fairly rare. Only 2 to 4% of white blood cells are eosinophils. Basophils, these purple-blue colored ones, are even less common, less than 1%. They're combating allergies and parasites again. Plus, they carry histamine and heparin to a wound infection. I'm guessing you know that word histamine, but maybe you've heard it used as an antihistamine. If you are having an allergic reaction, your airways might be swollen up so you're not getting air in. You might take an antihistamine to control that. So you might guess that a histamine then would cause that swelling, would cause fluid to go into a tissue so that it swells up. You might wonder why it would do that. Well, that's going to be a topic for the lymphatic system. There is a good reason why it would do that, and it has to do with how your body protects itself. Heparin, if you remember heparin, I doubt you do. But it was what was lining those little capillary tubes that you put your blood in. And heparin is an anticoagulant. It's a clot buster, if you will. It prevents clots from forming. So in those little capillary tubes we use during the blood lab, you put blood in there. You didn't want your blood to coagulate. You wanted to be able to separate the white cells from the red cells from the plasma, so it had heparin in it. Well, some of these little packets on a basophil carry heparin to a wound site. Again, you might ask, if I'm bleeding, why am I dumping chemicals in there that are going to keep me bleeding? And I'm guessing some of you might figure it out. And in fact, it's to flush that wound out. So when you cut yourself, you might be injecting bacteria, viruses, who knows what kind of pathogen into the wound. And so by keeping that wound bleeding for a period, not forever obviously, but for a period, your body is attempting to flush that out, that pathogen out of that wound. And so heparin is carried to those wounds by basophils. That's what some of these packets are on the surface of a basophil. All right. Lastly, the second to last, the agranular lymphocytes, monocytes and lymphocytes. And monocytes are the biggest and lymphocytes are the smallest. You can tell a lymphocyte because it's almost all nucleus. When you see it under a microscope and it's been stained, there's very little cytoplasm, almost all nucleus. Monocytes are the biggest one. And monocytes are white blood cell eaters. These are what formed foam cells in that description of atherosclerosis. They're floating around your bloodstream. And if there's too much fat, too much LDL cholesterol, these will actually eat it, turn into foam cells, embed in an artery wall, release growth factors, the wall thickens, the collagen breaks down, the wall hardens, a pulse of pressure comes through the heart. That now inflexible artery is then forced with this pulse of blood. It cracks in response, internal, inside the vessel, and a clot forms, and you've got a problem, a heart attack or a stroke. That's the foam cells formed from these. You're also going to see this word macrophage come up from time to time. And this is really a cell-eating cell as well, just like the neutrophil is. So you can go back to that video of an amoeba eating 2 paramecia, and that's a good image to have of a monocyte as well. So lymphocytes are the smallest, as I mentioned, white blood cell, and they're the second most common type of white blood cell. And they attack a broad range of foreign cells and abnormal cells from our own body. So, cancerous cells, that kind of thing. Yeah, our body has built into it the ability to sort of take care of cancerous cells. There's a school of thought within medicine out there that sometimes we're too aggressive with our treatment of cancer. And in fact, if we would just let our bodies do their own thing, more times than not, our bodies would take care of the cancer before we have to go through any chemotherapy or radiation therapy that can damage many other cells in our bodies and have many deleterious effects as well. The problem is, how do you know when that is? How do you know when your body's going to beat a cancer? And if you're wrong, is the cancer going to get away and kill you before you figure it out? So anyway, it's an interesting discussion. Wanted to mention one more thing about monocytes, and that's mononucleosis. And I'm guessing a number of you have had mono, and it has that moniker of being a kissing disease. And actually, that's true, because one of the best ways to get mononucleosis is to exchange saliva. There are other ways, drinking off a can of pop that somebody else has drank off. Number of ways you can do it. But basically, mononucleosis really doesn't have anything to do with a monocyte. It's actually a lymphocyte that gets infected by an Epstein-Barr virus. And it turns these lymphocytes into something that looks like a monocyte. When the disease was first described, people saw these abnormal lymphocytes, thought they were monocytes, and called it mononucleosis. Upon further research, they figured out they weren't monocytes. They were atypical lymphocytes, and then we have a disease with sort of a misnomer for a name to it. But you can get those Epstein-Barr viruses that kind of hang out dormant in the salivary glands of somebody, and then if they get stressed and run down, they can sort of flare up and be passed to somebody else and cause mononucleosis. So it is kissing disease, but it doesn't have anything to do with monocytes. It's lymphocytes. That's the problem. The last topic here is platelets. And so we go back to these stem cells that all blood cells form from. This is a stem cell that comes from the long bones. We talked about this on Wednesday. Basically the pelvis, the vertebrae, all these leg bones, arm bones, will not every bone in your body, but those bones will produce stem cells. And these stem cells can become red blood cells, any of the 5 types of white blood cells. And then lastly, this megakaryoblast, which basically blows up into pieces and forms platelets. So if we go back to this image that we talked about before when we were talking about the proteins that are in plasma in blood. So basically right here, you can see a poke. You've got a hole that is created in a blood vessel. So I kind of jumped ahead when we were talking about dominoes and all the 11 conversions of one protein to another that have to occur before you get fibrin like you see in the picture here. That whole thing starts, this is probably pedagogically not a good way to do this because I'm going back to the beginning. That whole process starts right here when that blood vessel is punctured, and the first thing that goes to that puncture are platelets. They congregate and form a plug and stem the flow of blood. And then they release a clotting factor that starts this whole progression. Ends up with a fibrin clot and captures blood cells just like a spider web would. My daughter and I were watching The Hobbit lately. There's always the spiders. There's Hobbit and Lord of the Rings. Always these nasty spiders, spinning webs. And yep, that's what we're looking at, except they're not catching hobbits, they're catching red blood cells. And of course, we mentioned that vitamin K is important in forming these proteins and in this whole process of blood clotting. So those are the formed elements, the last of the formed elements, the solid parts of blood. Red blood cells, white blood cells, and platelets. And I hope you guys have a really nice spring break. And when you get back, we will pick it up with the lymphatic system and go on from there. And I'm going to see if I can get this thing stopped. My machine doesn't behave well and won't let me pause the video very well.